Hi, my name is Chris Conley, Director of R&D at Streck, and I'm here today to tell you about our automated biotech data enhancement project. However, before we jump into talking about the project, I wanted to take a few minutes to tell you a little bit more about Streck. You may or may not know that Streck is a local biotech company here in La Vista, Nebraska, and we have just over 400 employees. Streck has been around since 1971, so we've been in business for over 52 years now. And our core technology was really founded on principles of cellular stabilization. And we've used that technology to develop many products over the years that service the clinical laboratory space, including quality controls for hematology analyzers. So if you go to the clinic to have your blood drawn and you're getting your hematology work done, so a complete blood count, for example, our controls would test the hematology analyzers to make sure they're running properly before your sample or a patient sample ever touches the instrument. Over the years, we've expanded on that technology to develop blood collection tubes that effectively stabilize whole blood after time of draw and allow it to be transported to central labs for analysis. We've also expanded our controls, our quality control materials for molecular diagnostic instrumentation, and we've developed assays for different instrument platforms. And that's where we're interested in talking to you a little bit more about this project and getting into why improved data analysis methodologies and automated data analysis methodologies are really important to our R&D division. So with that, we're going to dive into a little bit more detail throughout the slide deck, and I'll first highlight the problem, followed by the project statement and our preferred dev environment. And then last, but certainly not least, we'll introduce you to the fantastic team we have on this project from Streck. So I want to elaborate a little bit on why the team at Streck is so fantastic. We have the same mission. We're all working together to develop products that reach the clinical laboratories, and they essentially help people live healthier day-to-day lives. Our products impact their well-being. So it's critically important to us that we ensure whatever products we put on the market perform as they're expected time and time again. So for us, it's not just about the authenticity and accuracy of the science behind these products. It's about building a relationship with our customers that buy the products and the patients that benefit them, and essentially that's trust. However, the challenge that we consistently face is that we grow as the market evolves. So as new instrumentation and new tests are brought to the clinical market space, we're developing new products to meet the needs of our customers. With that evolves a lot of data collection, and of course the analysis that goes along with that. When we collect data on these products, we're typically ensuring that those products meet a specific criteria of performance. We collect hundreds to thousands of runs on that product over time to make sure the product's stable and to make sure it's repeatable and reproducible. To do that, with whatever instrument system the product is designed for, we have to analyze it typically on more than one of those systems. That takes a lot of time if you're doing it manually. Now, compound that by considering that we develop products for multiple instruments and multiple tests. So as an R&D group, we're looking for a way to automate that data extraction and analysis in a way that doesn't conflict with the quality and the time that we spend to ensure everything is accurate and precise, but allows us to move at a much faster and more efficient pace that ultimately allows us to divert our resources to doing more data collection and scientific discovery. So with that problem in mind, Streck's Research and Development Department is looking to improve upon our manual methods for extraction and analysis of data from various instruments. And what we're hoping to do is find an automated way to do that that allows us to become more efficient while keeping the same level of quality that we expect of our work. So with that in mind, there are four main goals of the project. One, and most obvious, is automate the data extraction. We want to find a software tool that will be compatible with multiple instrument platforms and will allow us to rapidly pull the data as it's collected from the system. We then want to be able to precisely and specifically look at these data sets so that we can rapidly analyze specific parameters, so parts of the data, or collectively analyze the whole of the data, and then rapidly generate a report that we can provide to show the output of whatever data collection that we're currently doing at this time. We then need the capability to store all of the raw data files within our electronic laboratory notebooks so that we're able to go back and review any data should questions ever arise. And then we need this software tool to be compatible and integrate with multiple devices and systems, because the unique thing about all of these devices and systems is they can have slightly different file types that they export following data analysis. Taken in total, however, by automating our ability to extract and analyze the data, we can gain a significant amount of efficiency back in time. And that is most critical to us when we're working in a very fast-paced and highly regulated environment. So as a research and development group that's primarily working in the lab on the bench and doing a lot of wet chemistry applications and collecting that data, we started talking about this problem and recognizing that we wanted to become more efficient. So as scientists, we decided we were just going to start looking into things ourselves. And we wrote some code using Python, developing some simple programs that really ultimately helped us, but it wasn't good enough and frankly isn't our expertise. So we started consulting our IT group, maybe something we should have done early on. And our IT group said, hey, yeah, I think this is something that we can work on together, but we also have this opportunity that is available, and we can work with UNL to develop potentially a software program that would really meet our needs and be much more effective than some of the work that we were just doing kind of freelance in the lab. So as we started talking to our IT team, they suggested that we could use React as our front-end library. We could use C# as our back-end language. Database could be the Microsoft SQL server, and then we could host everything in Azure. Now, I don't know what any of that means. So that's where I'm excited to entrust you guys and let you know this is kind of what we're proposing, but we're very open to hearing your feedback and how you'd proceed with this project and what you can develop for Streck to bring to us something that's ultimately going to improve the quality and our ability to collect and analyze data. So as we look into kicking this project off, I wanted to take a moment to introduce you to our leadership team. First and foremost is our Chief Executive Officer, Connie Ryan. You may not know, but she is the daughter of Streck's founder, Wayne Ryan, and she started off in medical technology, and she's worked through various areas of the company, and now she leads us all and is constantly looking for ways to support us, to allow us to continually improve and help us do our jobs better so that we can produce more effective products for the clinical market. Our project sponsor, Chad Lynch, who's our Vice President of Information Technology, he, along with some of the project team, really brought this idea to our attention, and he's going to be here to help support and make sure the project team has everything they need to be successful. Next, you have myself, Chris Conley, as well as John Scholl. We're both directors of R&D here at Streck, and between the two of us, we oversee multiple divisions and multiple product lines, and we have an excellent idea for the various type of data files and instrumentation that we will routinely use, and we can serve as advisors on really how to make this software tool most effective for our day-to-day use, and we'll help work directly with the project team also to support and give them the tools they need to be as successful as possible with this project. So last and certainly not least, I want to introduce you to our project team. It's our crack experts who on the Streck side are going to help make this project a success. and who know the inner workings of Streck as well as the needs of the project and will communicate with you on a regular basis. So on our IT side, we've got Caleb Noonan, Scott Michaelis, and Priya Premnath. On our R&D side, we have Ben White, Shay Lott, Carl Bisselou. So with that, we'll let you get off to the races. We're excited to work with you on this project and see what you come up with. Look forward to being in contact soon, and good luck.
Where is John? Hi, John.
Hi.
Would you like to introduce yourself to the UNL students?
Yes, my name is John Scholl. I'm an R&D director here at Streck. As we are looking forward to working with the group as we've grown over the years. I've been here for 36 years. As we've grown, I've seen a lot of need for better methods of data analysis and how to collect the information and put it together in a more straightforward format. We look forward to seeing you.
Hi, Chris. Do you want to introduce yourself to the students?
Sure. I'm Chris Conley. I'm Director of R&D at Streck. I'll be one of your project sponsors, and I'm looking forward to seeing all of your success.
What are you doing?
Pipetting water.
Sounds fun.
Yeah.
Sounds important. Hey, Ben. Do you want to introduce yourself to the students?
Yeah, my name's Ben. I'm a R&D associate scientist, and I'll be the primary lead for R&D on this project, so I will look forward to working with you.
Caleb, don't you work in IT?
I do.
What are you doing here?
Science.
What kind of science?
Whatever this is.
Hello, I'm Chad Lynch. I'm the Vice President of IT here at Streck Biotechnology. On behalf of the IT team, we'd like to welcome you as a partner on the Capstone project, working on a application for research and development project here at Streck. So on behalf of the team, I'd like to introduce our other members that you'll be working with during this project. Caleb?
Hi, I'm Caleb Noonan. I'm a software engineer. I'll be the tech lead, and I'm excited to work with you.
Scott Michaelis, IT App Dev Manager. Excited to work with you.
Hi, I'm Priya Premnath. I will be the project manager for this project. We're super excited to welcome you to our team.